So I'm going to talk about next generation DNA sequencing. An example of next generation DNA sequencing is Illumina sequencing. I am now going to outline the main steps in Illumina sequencing. Step 1. Prepare genomic DNA sample. DNA is randomly fragmented and adapters are ligated to both ends of the fragments. Step 2. Attach DNA to surface. In this step, single-stranded fragments randomly bind to the inside surface of the flow cell channels. Step 3. Bridge amplification. Unlabeled nucleotides and enzymes are added to initiate the solid phase of bridge amplification. Step 4. Fragments become double-stranded. The enzyme incorporates nucleotides to build double-stranded bridges on the solid phase substrate. Step 5. Denature the double-stranded molecules. By denaturing the double-stranded molecules, we are left with single-stranded templates anchored to the substrate. Step 6. Complete amplification. As a result of the steps so far, several million dense clusters of double-stranded DNA are generated in each channel of the flow cell. Step 7. Determine first base. This is the first chemistry cycle. To initiate the first sequencing cycle and add all 4 labeled reversible terminators, primers, and DNA polymerase enzyme to the flow cell. Step 8. Image first base. After laser excitation, capture the image of emitted fluorescence from each cluster on the flow cell. Record the identity of the first base for each cluster. Step 9. Determine second base. This is now the second chemistry cycle. To initiate the sequencing cycle, add all 4 labeled reversible terminators and enzyme to the flow cell. Step 10. Image second chemistry cycle. After laser excitation, collect the image data as before. Record the identity of the second base for each cluster. Step 11. Sequence reads over multiple chemistry cycles. Repeat cycles of sequencing to determine the sequence of bases in a given fragment, a single base at a time. 12. Align data. It is necessary to align data and further comparing the data to a reference to identify different sequences.